Hi everyone, my name is Caroline Maro. I'm a PhD student at the School of Plant Sciences and I'm doing my research in the Anode Lab and the ARID Center. Today I'm happy to give you my lightning talk about my summer research experience entitled Enhancing Tepary Performance via Microbial Consortia in Productivity and Yield Perspective. So by introduction, climate change threatens food security by rising temperature and drought, as you can see from this picture, which causes food insecurity. One of the solutions is obtaining nutrient-dense crop that thrives these climate change impacts. And one of them is the tepary bean, and these are different photos of tepary bean varieties. And here you can see how tepary bean grows well under a very, very dry condition. Harnessing the potential of the tepary bean requires eco-friendly strategy to enhance its productivity under this stress environment. And there is where the curiosity of my research came. So I asked myself, what if the adaptation and resilience of tepary bean comes from the eco-friendly bacteria that resides inside its seeds? Looking at the importance of seeds for plant fitness, reproduction, and perpetuation of generation from one generation to another, that builds my curiosity of characterizing the bacteria that resides inside the tepary bean seeds and study how they impact tepary bean growth and resilience. And this is one of the plate that I characterized during the early stage of this research. So to answer these questions that I have, I did characterization of the tepary bean endophytic bacteria from the seeds, and I found out like 4 different species inhabit tepary bean seeds. I went further to look if this bacteria has some functional traits of making nutrients available for tepary bean as tepary bean grew in a nutrient depleted arid area. And I found out that they have ability, some of them have ability to solubilize phosphate. They have ability to chelate iron as it's indicated by the yellow halo in this iron assay. They produce ammonia, all of them, which is an important source of nitrogen for plant, but none of them produced the indole acid hormone. So after I found this, I went further to ask myself, if I bioprime, if I treat my seeds with individual inoculum and plant them, would the inoculum enhance the seedling growth? And I found out, yes, they do enhance the seedling growth of tepary bean. As you can see, this is one of the treatment that I had. It was micrococcus. Compared to its comparative control, you can see there is a difference between these two seedlings growth in terms of root, in terms of above ground. So I went further to ask myself, are they also doing the functional traits when they're in consortia or in combination? So I characterized the functional traits for multi-species consortia, and I found out, yes, they do solubilize phosphorus. So these are 2 different consortia. Some of them, they do produce the siderophore, as well as all of them produce ammonia, but again, none of them produced indole acetic acid. So like the way I did the experiment to see their impact on tepary bean growth, seedling growth, I went further to look at their impact on this tepary bean performance, and this was the basic of my summer research. So I asked myself, what are the effects of these microbial consortia on tepary bean performance? How do they affect early season productivity? How do they affect seed yield at harvest, as well as how do they influence root mass, which is a key component of plant resilience under stress? So I hypothesized that my microbial treatment in consortia will affect productivity, will impact seed yield, will impact root weight, as well as seed weight, which is an indication of grain quality. And I predicted that the plants that will be treated with my microbial consortia will have higher productivity than control, will have greater seed yield than others, and the control, as well as will have increased the 100 seed weight, which is a measure of quality or seed size, as well as they will have higher root mass than the control. To answer this, what I did, I bioprimed my seeds with different combination of consortia. I had like all these combinations, and this is how I did it. I treated my seeds with different bacteria and I incubated them for overnight before I took them to the field. And this is the early growth stages of my tepary bean under the field experiment. And I conducted the field at Maricopa under randomized completely block design with 4 replication each, having the blocks as my random factor and the treatment as the fixed factor. And here is the late stage of the tepary bean after I conducted this experiment. And later I measured the productivity index, 100 seed weight, yield, and root weight. And this was the day I was collecting the data, and you can see so many pods have been formed. After that, I did the statistical test whereby I conducted analysis of variance to see the impact of the variation between performance of the tepary bean if it's just coming by chance or were impacted by my treatment, as well as I represented my data in least square mean to see which consortia performed well for the variables that I assessed. So here is a result of productivity index, which include the early germination and early growth of the plant. And I found out that there was really statistical significant variation between the controls and the treatments that I applied with these 3 consortia of Acinetobacter plus Cochlear species 1, Acinetobacter and Micrococcus, as well as Micrococcus and Cochlear species 2, performed very, very well compared to other treatment as well as control. The combination here, you can note, of all 4 species didn't perform well as compared to these 2 consortia combination. The yield also were highly impacted by my microbial treatment, with, again, my 3 consortia performing very well compared to the control here. And I had also Cochlear species 1 as a single inoculum performing well. I included this because I missed this treatment during my last field experiment due to slow growth of my inoculum, the time that other inoculum were just already at exponential phase to be treating the seeds and take to the field. 100 seed weight were not statistically impacted by my bacteria consortia. As you can see, there was no differences in performance between the controls as well as the treatment. Similar results also obtained for the root weight, where I observed there's no difference in performance of root weight between the controls as well as the my microbial consortia treatment as signified by the statistics. So in conclusion, the focal microbial consortia improved my plant performance by enhancing early season productivity and yield, as we saw from the results. The root weight and seed quality were not impacted by my microbial consortia treatment. 3 species of Acinetobacter plus Cochlear species 1, Acinetobacter plus Micrococcus, as well as Micrococcus plus Cochlear species 2 showed a very strong synergistic effect. And these consortia represented the functional traits, if you remember from the functional traits assays, such as phosphate solubilization, siderophore production, as well as ammonia production. So what I'm planning to do next is to evaluate the nutritional quality of the tepary bean that produced in this field experiment to see if my consortia impacted also nutritional quality. The relevance of this research is to ecosystem genomics is just showing us from the characterization how the genomic and functional trait diversity within the seed bacteria consortia influence plant productivity in an ecosystem. Apart from this summer research experience, I applied for a fellow to be a fellow in a global food PhD platform, and I was selected as part of the members, and I attended the Global Food PhD workshop in South Africa. This is sponsored by the University of Montpellier in France and the University of Laval in Canada. The platform is designed to foster the solution to food insecurity through resilient crops that can thrive despite these climate changes, as well as combating chronic human disease. And this is where my climate resilient tepary bean fits in. And I attended this workshop and I presented my research on endophytes for the future in a perspective of what tepary bean tells us or shows us about thriving climate change by having the seed endophytic bacteria within it. My advice to the next cohort: always stay curious and think across scale. don't just focus on one aspect of your research, like maybe the genomic data only, but go further to ask yourself how those genetic functions shapes community interaction, the environment, and the real world ecological outcomes. Embrace any complexity that you find in your research, because there is where most meaningful discoveries are coming. Like the way I show you, my 4 bacterial combination didn't do very well compared to only 2 consortia. Look for and accept for opportunities to build your career and professionalism, such as attending workshops and conferences. This will help you grow academically. I would like to acknowledge my advisors, Dr. Betsy Arnold and Dr. Duke Polai, for their mentorship and support that enabled me conduct this research. The members of Arnold Lab for their support during conducting of this research and all the experiments that I did and data collection, that they supported me, and different sources of funds, including the ecosystem genomics that provide part of the funds that enabled me conduct this research. These are the references that I used in my work, and I am glad to take any question.